My name is Stephanie Forkel and I'm presenting the paper Stroke Disconnectome Decodes Reading Networks. The innovation of written language is a pivotal hallmark in human history. Mastering reading and writing requires many years and the interplay of several cognitive and linguistic processes. Cognitive functional neuroimaging has been around for over 30 years and has shed light on the brain areas relevant for reading. Seminal work on the functional brain activations has shown that reading selectively activates bilateral temporal occipital areas, including the left visual word form area and right occipital areas. However, reading proficiency modulates a more comprehensive left hemisphere language network, suggesting that literacy acquisition is tapping into the extended language circuitry in the brain. This network includes the left temporal cortex, premotor, and supplementary areas. The inferior frontal gyrus harboring Broca's area has been associated with various linguistic processes, including phonological segmentation, syntactic processing, and unification. Literacy, therefore, requires a network of language processing, auditory, and visual regions to interact. These relevant regions are distant from each other and as such rely on a network of connections. While white matter studies sporadically highlight parts of brain networks necessary for reading, their relationship with the networks whose damage will contribute critically to reading impairment is not well defined. A potential way to fill this gap is to take advantage of recent developments in functional structured connectivity coupling that provides methods to decode functional fMRI activations based on the pattern of brain disconnection derived from stroke, known as the disconnectome. Briefly, we used previously published data as shown in the gray boxes for a functional reading task and the disconnectome maps to calculate the group level statistical map for reading and use the disconnectome to decode the critical white matter network. The reading task activates the bilateral network of regions in the frontal temporal occipital lobes and subcortical areas. Mapping the profiles of disconnections identified reading clusters in the occipital lobe centered around the vertical occipital fasciculus and another white matter cluster located in the short U-shaped fibers connecting the precentral to the middle frontal gyrus. A typical lesion was identified with a disconnection profile that could explain 33% of the variance in the reading task. Accordingly, this lesion will likely disconnect the white matter pathways feeding into the activation pattern of reading. The lesion most predictive of a reading deficit impinges on the left temporal occipital and inferior parietal gyri. Such a lesion should be considered as rare, occurring in less than 1% of the stroke population, and may explain the rare occurrence of pure alexia in stroke. Now, this method reveals the white matter circuitry connecting activated areas and typical lesions that should interrupt the circuitry, thereby identifying the critical white matter circuitry for a specific cognitive function, in this case, reading. This novel framework can systematically be applied to bridge the gap between the neuropathology of language and cognitive neuroscience.